Avrobio is a lentiviral-based ex vivo gene therapy company focused in rare disease. Our 4 pillars are on the screen. We're very proud of the team that we've built. We think we have significant operational skill sets in scaling up and running trials for ex vivo therapies. We have a 3rd-generation optimized lentiviral platform, and we have significant investments in vectorology to continue to advance. And most excitingly, we're in the clinic, is that we recently have treated our first patients in 2 indications, Fabry's disease and AML. And following these 2 clinical stage assets, we have what we think is a very unique and differentiated pipeline, which leverages everything that we've done. So the same cell types, the same lenti, just a different transgene. What we're really most excited about is the team that we've built. And what we've tried to do is to co-culture experience in cell and gene therapy with rare disease. And some real cell and gene therapy veterans on the left with some rare disease industry leaders on the right. So I think just some of the brag points we could reference is that on the cell and gene side, probably about 1.4 or 1.5 million patients have been treated in the world with cell therapies. 1 million of those are from organizations that we've led. Kim, who leads our operations, ran a team of 50 in process development in Lonza for about 15 years and have scaled up every kind of cell therapy you could imagine. And Chris, prior to us, spent a dozen plus years as chair of regenerative medicine bioprocessing at UCL. And beyond that, we have other team members. We've really consciously tried to build that operational leadership in cell and gene, but then graft on the rare disease side. And not everybody here has worked with Arthur and Shire, but many have. So Deanna, right? You're feeling nostalgic, Arthur? So Deanna was head of BD&L for rare disease for Shire. Narissa has done the gamut of rare disease, working most recently as medical affairs and clinical lead at Zafgen, but prior to that in senior roles in Shire and Enzyme and Alexion, and similar to Catherine. And we also have the head of toxicology preclinical from Shire. So we've really consciously tried to meld these 2, cell and gene with rare disease. And the focus of the company is listed here. So Fabry's disease, we've treated our first patient in January this year. We've recently announced an in-licensing of a very late stage and near to clinic opportunity in Gaucher's disease. We have one that we're not quite ready to announce, but we're very excited about our 3rd rare disease asset. And we're in the clinic as of literally a few weeks ago in AML for and also an ex vivo gene modified technology. So to start maybe a little bit about Fabry's and then the standard of care. So Fabry's disease is a monogenic rare disease. The patients can't express an enzyme. Alpha-Gal A leads to an accumulation of a fatty substance, GB3, and has a very serious deleterious effect to the patient. His life is cut short, but the life that they live is often miserable with renal effects, cardiovascular effects, GI effects, pain, depression. So really affects the patient head to toe. The standard of care today is enzyme replacement therapy. So the Shires and Genzymes of the world sell $1.2 billion a year of enzyme replacement at, I think it was just referenced, $250,000 plus per year for life. And if the enzyme replacement therapy was sufficient, that would be one thing. It would still be onerous because the patients succumb to biweekly infusions for life, which is, of course, takes over their life to an extent. But we also think that although it's the right enzyme, the pharmacokinetics are suboptimal. Is within a few hours of the infusion, the enzyme is gone from the blood. So the whole premise of ex vivo gene therapy is with an outpatient conditioning, so a mild ablation, one single infusion could deliver 24/7 expression of enzyme for long-term, hopefully permanently.
And in the ongoing trials, are patients discontinuing ERT, and how is that background of standard of care being incorporated?
So the first phase 1 trial has been designed conservatively because, of course, it's a safety trial and it's the first Fabry's disease gene therapy trial, and I think perhaps the first adult LSD trial. And so the protocol is designed where the patients are titrated off enzyme replacement therapy, the gene therapy is applied, but ERT will be reinitiated after 28 days. And that may be revisited as more experience is provided. But right now, what that trial allows us to do is, first of all, measure safety, measure engraftment of the cells, determine with confidence that this partial ablation conditioning regime is effective, and look at the expression of enzyme. But beyond that, some of the functional parameters are co-founded by the ERT.
And what will you be looking for in terms of efficacy, and what would you expect based on your preclinical work?
So for this phase 1, first in humans, the surrogate for efficacy will be enzyme expression primarily. Have the cells engrafted? Are they expressing enzymes in physiologically relevant concentrations over time? Most importantly, we're in late stages designing a phase 2 clinical trial, and the phase 2 clinical trial will actually be staggered start prior to the end of the phase 1. And this trial will more thoroughly address what we would call functional endpoints. So beyond just looking at enzyme expression, substrate reduction, safety, some of the endpoints that are required for eventual regulatory approval.
Have transplants been utilized in Gaucher's or Fabry's or any of these lysosomal storage diseases?
So to the best of our knowledge, there's no clinical trial for Fabry's disease with bone marrow transplant, allogeneic bone marrow transplant, and we don't even know of any case studies. And we assume that's really just because of the risk profile of allogeneic bone marrow transplant. There have been mice studies which would suggest that it would be promising, but I guess it's just the risk-benefit ratio. Gaucher's disease, it has been. I think the first was 1982. And with significant success is that you can see multiple patients have been treated since that time, and you can see spleen and liver reduced to normal size, and platelet counts and other measures return to normal. So we see that as obviously as a nice de-risking surrogate.
Moving on to the oncology side of the company, maybe just describe the role of IL-12 in cancer, and why gene therapy is an attractive approach for cancer.
Sure. So what the technology is, just to orient you, is an autologous process where we take the patient's blood, we select out the blasts, so rather than T cells, the cancer cells themselves, the neoplastic cells, transduce these neoplastic cells to express IL-12, and then reinfuse back into the patient. And so the premise is really to benefit from two factors. One, to help the CTLs target all of the multiple neoantigens. If you have a cancer that's heterogeneous, where you can't just lock on to, say, a CD19, for example, then we think that this would be a benefit to target all of the multiple neoantigens. But the idea of using IL-12 to prime that immune response is attractive. And an IL-12 is well recognized to be a potent anti-tumor cytokine. Its challenge is that it's toxic when administered systemically or administered as being engineered onto a CAR or a TIL, because really that's just another way of delivering it systemically. So this premise is to deliver IL-12 in sufficient concentrations locally to the lymph node to create a spark of an immune response, and then these very cells would be killed by that immune response. But the hope in what we saw in preclinical is it's enough to trigger this potent immune cascade.
And so you don't think there's any risk of constitutive expression of IL-12 or anything systemic or too much IL-12?
Well, so these are non-dividing cells delivered locally. So the whole premise of this approach is to avoid that systemic toxicity. So we would hope and expect not.
Any questions from the audience?
Hi, Donna Armentano. So I was wondering if you were able to look at levels of enzyme in the kidney for your Fabry's studies, and how that would compare to ERT, and if you could comment on potential benefit for the kidney in Fabry patients.
So in this phase 1 trial, we are not. In the phase 2, we're looking at doing biopsies, renal biopsies, skin biopsies, a few different ways to address that. So we don't have an answer right now. I think the data for ERTs on kidney function is mixed, and I think it was a tough job for regulators to sort through it because it wasn't as pristine as one would hope. So, our hope and belief is that we'll be able to demonstrate a functional benefit for kidney. What's interesting about Fabry's disease is that particularly the US FDA seems to be orienting more as a primary endpoint towards a patient-reported outcome GI-related endpoint. And the premise is that they're no longer satisfied with enzyme expression substrate reduction as an approvable endpoint, but the recognition that something like kidney is very long and arduous to demonstrate in the context of a clinical trial. So it may be a secondary endpoint, and it could be that something like a PRO GI reported, at least for FDA, would be primary.
Any other questions from the audience? Maybe one more on Fabry. Where do you think the enzyme level that you'd be looking for would be? And just given that the profile is continuous, do you think that changes? Maybe how to think about that?
Yeah, I don't think we have. We don't really look at the enzyme replacement distribution or kinetics as something that we can learn much from, because there's this huge bolus of too much and then it's gone, too much and then it's gone, and it's unclear how much are taken up from the cells. So even area under the curve is not really that relevant. I think we're heartened. I think Arthur made the point that it's important to select a clinical indication where you're not asking too much of the modified cells. And there is some research in these types of lysosomal storage disorders that even just delivering a few % of the enzyme is enough to impact the disease. So we don't really know what the optimal level needs to be, but we think that the real benefit is that 24/7 expression. And even if the area under the curve doesn't match the bolus of ERT, we think it can still be disease modifying.
Great. Well, thank you very much, Geoff.
Thank you.